Good afternoon. Myself Mr. G Kulkarni. Today I am going to deliver a lecture on lipid oxidation as a part of paper Metabolic Pathway for BSc Second Year Biotechnology. As all of you are aware, different biochemical reaction takes place in our body. It is a type of oxidation which brings about oxidation of the lipid, and huge quantity of energy is obtained via lipid oxidation. This metabolic pathway, fatty acid oxidation, is carried out in bacteria as well as in plant as well as in higher animals like human being. We are going to study the beta oxidation of the fatty acid. The fatty acids are metabolized in mitochondria, and there are different steps involved during the oxidation of the fatty acid, and huge quantity of ATP, near about 130 ATPs are obtained via this oxidation pathway. So there are different steps involved in the process of activation. The name beta oxidation, it is called beta because the oxidation actually takes place from the beta carbon atom. So in this slide, all of you observe there is mention of alpha and beta carbon near the carbonyl carbon. So from this beta carbon position, actually the process of cleavage or oxidation takes place. So it is called as beta oxidation. This oxidation discovered by scientist Noop in 1909. He did the experiment on experimental animal like dog, and he fed the carbonated and phenyl containing derivative to the dog and found the content of the digestive system by post-mortem analysis, and he surprisingly found that the 2 carbon less units are found during the digestion of that lipid. So give the name beta oxidation. Before actual oxidation inside the mitochondria, it is necessary to activate the fatty acid. It is called as fatty acid activation. It is a reaction that is catalyzed by an enzyme, acyl-CoA synthetase, and this activation reaction is carried out on the surface of the mitochondria, which utilizes the expenditure of the ATP molecule, which is split into AMP and PI. So 2 high energy bonds are utilized during the activation of the fatty acid. So it is activated form of fatty acid is called as acyl-CoA form. Once the fatty acid get activated, the acyl-CoA form of the fatty acid now enter inside the mitochondria. But those fatty acids which are having carbon length more than 8 carbon, they are not easily transported inside the mitochondria for further rounds of the oxidation. So for their transport, the transport mechanism required is called as carnitine system. This slide shows the difference between acetyl-CoA and acyl-CoA. Generally, acetyl-CoA like molecules, they are found in the as a part of the reaction of intermediates of the TCA cycle when pyruvate is converted into acetyl-CoA. So one may get confused between the structure of the acetyl-CoA and acyl-CoA. Acyl-CoA is the activated form of the fatty acid in which R stands for number of the CH2 groups which are present linked with the methyl group. But in the structure of the acetyl-CoA, the R stands for only the methyl group. So this is the difference between acetyl-CoA and acyl-CoA. So if we take the example of the 16 carbon palmitic acid as a fatty acid oxidation example, the R stands for remaining whatever the 14 carbon atoms which are present in the form of CH2 length, and earlier CH2 and 1 CH3 contribute for next 2 carbons. So total 16 carbon fatty acid oxidation, palmitic acid we are going to take into consideration for this example. Now this is accomplished to the beta oxidation, which activation takes place on the surface of the mitochondria, but actual oxidation takes place inside the mitochondria. So how lipids move into the matrix? As I mentioned earlier, those lipids having carbon atom length more than 8 to 10, they are transported via the carnitine shuttle system. This diagram shows how the activated form of the acyl-CoA form of the fatty acid is transported via the carnitine. It is converted into acyl-carnitine form, and then via carnitine transporter system, it is also called as translocase system, the activated fatty acid then inserted inside the mitochondrial matrix for further rounds of the oxidation. And again carnitine release free in the cell cytoplasm again to acquire new incoming form of the fatty acid. So this diagram shows how carnitine molecule act as a transport molecule for transfer of activated form of the fatty acid from cytosol to the mitochondria for further rounds of the oxidation. So this is the general structure of the mitochondria. The beta oxidation refers to the fact that the reaction takes place at the beta carbon. So this diagram shows the location of alpha and beta carbon. From beta carbon, the fatty acid oxidation takes place and the chain is minimized by 2 carbon. So in each successive cycle, how the chain is minimized by 2 carbon? As we are taking the example of the 16 carbon palmitic acid, 14 carbons are formed when 2 carbons are minimized in one round of the oxidation. Now this reaction one by one we are going to study how the cleavage takes place. As mentioned, 16 carbon palmitic acid in front of us, how it is treated by the first step oxidation, which releases the FADH2. Oxidation is nothing but removal of H. Due to removal of H, the structure becomes trans delta to enoyl CoA. It is called trans because both the hydrogen positions are opposite to each other. Then second step is hydration, addition of water molecule. Again in third step by the enzyme dehydrogenase, removal of H molecule, then formation of the NADH, and lastly the thyrolysis process, which minimize the chain by 2, and the end product is acetyl CoA in the 2 carbon structure. Remaining chain is minimized by 2, is contain 14 carbon structure. So this is the one round of the oxidation in which one NADH, one FADH2 and one acetyl CoA is formed. As we are taking the example of palmitic acid, there is requirement of the 7 rounds of the oxidation. This is separately, each reaction is explained. And as compared to this 7 round, how many NADPH and FADH2 they are liberated and how they count for the formation of ATP, this isometry shows. As per one round, one acetyl CoA molecule is liberated. The total 8 acetyl CoA are produced in 7 rounds of the oxidation. So when one acetyl CoA enters the TC cycle, 12 ATPs are produced. For 8 molecules, 12 into 8, 96 ATPs are produced. For one round of the oxidation, one FADH2 and one NADH are generated. So for 7 rounds, we are counting 7 into 2, 14 ATPs for FADH2 and 7 into 3, 21 for NADH plus H plus. As these are the reducing power, they are generated and transported in the electron transport chain for generation of ATP. So net gain of the beta oxidation of fatty acid is total 131. And as we mentioned earlier, in activation of the fatty acid, 2 high energy bonds are utilized. So net gain is 129 ATPs are generated when single 16 carbon palmitic acid is hydrolyzed. So this is about the how the beta oxidation 16 carbon palmitic acid chain is hydrolyzed inside the mitochondria as one of the biochemical reaction. Thank you very much.